efficacious level of intervention in these things might in fact be made not by assuming that everything should be down here on the spectrum of persuadability, in other words, addressed by typical tools by which we manipulate machines, today's machines anyway, but actually to really understand where cells and tissues and organs and so on lie on the spectrum. Because if they're not just here, and it's just an assumption that they're all here, then other tools, starting with cybernetics and control theory, but then leading to behavioral science and the kinds of things that are done there, and even more advanced things, might in fact be amenable. So in order to see how this shakes out, I want to start thinking about this fundamental problem, the self-assembly of the body. So we all start life as a collection of elastomeres. Eventually, we get this. This is a cross-section through a human torso. You can see the incredible order that is here. And the obvious question is, where does this pattern come from? And when I give this talk to school children, even 9 and 10-year-olds raise their hand. And they say, "Well, it's in the DNA. It's in the genome." That much is clear. So we want to talk about the fact that actually, if you read the genome, which now we can, what you see is that what it specifies are proteins. It specifies the molecular level hardware that every cell gets to have. It does not directly actually say anything about this, about the types of organs you're going to have, the symmetry plan of your body, any more than the genome of the termites or the genome of the spider tells you exactly what the structure is going to be here. These construction projects, very much like this construction project, is the result of really critical physiological software that runs on that hardware. And in practical terms, this means we need to understand, how do these cell groups know what to make? How do they know when to stop? How do we induce them to repair? And then as engineers, we would like to know, what else can we make them do? And this is a slide that I've shown a million times that reminds us that it's an amazing capacity, this kind of homeostasis, which not only builds a particular structure, but then is able to repair it, and most importantly, to stop when it has achieved the right journey in anatomical space, is really something that we are all shooting for in regenerative medicine. We would like to understand how these cells know that something is wrong here, and how they minimize the error, the delta between this state and this state, and eventually get up here. So the end game, as I've claimed before, for this whole kind of thing is something I call the anatomical compiler. What you want is that someday you can sit in front of a computer, draw the animal, plant, organ, or biobot that you want, and the system should compile that description into a set of stimuli that would have to be given to cells to get them to build exactly what you want. So we're talking about complete control over growth and form. The practical implications are enormous. If we had this, all of this would go away. Birth defects, traumatic injury, cancer, aging, degenerative disease. If we had a way of making cells build what we want them to build, this would all be a non-issue. Now, why don't we have this? Genetics and molecular biology have been going gangbusters for decades. Why are we very far away from this? And partially, I'm going to claim it's because people tend to think about things like this as some sort of 3D printer, some kind of thing that either arranges cells or micromanages gene expression or cell position or something. And I think that's the wrong way to think about this. I think what this really is a communications device. It's a translator between the goals of the engineer or the regenerative medicine worker and that of the cellular collector. And I think where we are today in molecular medicine is basically here. It's where computer science was in the 40s and 50s, where in order to get this machine to do something new, you had to physically rewire it. So there she is sitting there and altering the physical structure. And so today, CRISPR, so genomic editing, protein engineering, pathway rewiring, all of those things are focused, they're laser focused at the lowest levels of chemistry, trying to understand those kinds of details. So we're getting very good at this. We're getting very good at manipulating molecules and cells, but we're a long way away from this. So what I want to do in the rest of this talk is not just talk about the mechanisms that restore standard organs and standard tissues, which I've talked about many times before. I want to focus on what I call the creative plasticity of biological tissue. I want to see what else it's capable of making, because I think by understanding those things, we can shake ourselves out of the assumption that these things are a mechanical event that inexorably leads from the specification of the hardware to some sort of outcome, and really understand the quality of the agential material of life. It's not just complexity; it's actually decision making and problem solving, and that's what's going to lead to future medicine. So one of the reasons you need something like this is that this is the kind of model that is very popular as figure 7 of the cell paper, where somebody's done really hard work and identified all the molecular interactions. But this happens to be the specification of the eye field. Looking at this, it's very hard to first of all say what these things are going to build, and to then do the engineering of saying, okay, but if I want something slightly different, what part of this am I going to change? What we really want is something like this. I mean, this comes from 1980, but Jonathan Slack's algorithmic model of planarian regeneration. But these are extremely rare. Models that make it clear how it is that you would interact with this body of knowledge to actually fix something are really, really not common. So we need to understand how we can do that and go beyond the tales of the molecular hardware to something that we can use for interventions. So let's talk about now the kind of gaps that are in our textbooks. There was the two small clouds in physics when people thought that it was kind of mostly a done subject, turned out to be special relativity and quantum mechanics, and I think biology has way more clouds like this that are not apparent from the textbooks. So let me show you a few examples. So first of all, the standard story of morphogenesis of our body construction is that cells differentiate. In different parts of your body, cells will activate different parts of the genome. So they have different nuclei, they have activate different parts of the genome, transcribe different genes, and thus you have morphogenesis. Well, so let's see. So this thing is a single cell. Okay, so there is no cell differentiation as such. All the pattern is created within a single cell by the manipulation of physical forces that themselves are not encoded in the genome. This whole thing is one cell as well. This is an alga known as an acettabularia. Here's a stalk, some roots, this beautiful cap. It's actually very large. The whole thing can be 5 to 10 centimeters in size. There's one nucleus. Okay, so you're not going to be telling stories of differential gene expression in different locations of different types of cells and different types of the body. This whole thing is one cell, just one cell. And then another really critical thing is this non-genomic inheritance. So this is a ciliate. It's a little single cell organism with these little hairs on their surface. And these hairs are waving. And one of the things they do is they wave food into the mouth. And so they have this very coordinated flow that sort of wafts material into the mouth. So what Sonneborn and then later Besson and Frankel did was to cut out little squares of this surface, turn them 180 degrees, and put them back. Imagine doing that microsurgery on a single cell, okay, but they were able to do it. And what happens is then these cilia wave in the opposite direction, and they end up waving the food away from the mouth. And there's a paper with this beautiful phrase in it which says, this line, and it is a line of animals because actually when these things divide, they template off the cytoskeleton, and they make offspring that have exactly the same issue, this little square that's going in the wrong direction. So this is not genetic. There's nothing wrong with their genomics, but it propagates, and you can make permanent lines of these. And the beautiful line in that paper is that these strains of ciliates were always on the edge of starvation, and their normal genome was powerless to help them. Right? They were pushing half the food away from their mouth, and they have a perfectly normal genome that was making all the proteins that you want. Nothing wrong with any of them, no mutations, and yet profound differences in viability because this kind of thing is hereditary but not genetic. And Chris Fields and I reviewed some of these things in this paper. Okay, so what else can we say about genetics? Well, here are baby axolotls. Baby axolotls have little forelegs. Frog larvae do not have forelegs. In our lab, we make something known as a frogolotl, which is a combination of frog and axolotl tissues. You have the axolotl genome. So it's been sequenced. It's been annotated. You have the frog genome. Could you look at those genomes and tell me if a frogolotl is going to have legs? And if it does have legs, are those legs going to be made entirely of axolotl cells or frog cells? And the answer is no. We have no models that will allow us to make any kind of prediction like that. In fact, if we weren't cheating and comparing these genomes to creatures that we already knew what they look like, you couldn't even tell me what the baseline ingredients were going to be given the axolotl genome. We have no way of saying this is the thing that's going to come up. So here are some other limitations of the genetic information per se. Now, let's think about how many embryos are in what we conventionally see as an embryo. So you look at a blastoderm, there's 100,000 cells or something, and we say, oh, look, there's an embryo. What do we mean by saying that, by calling that collection of cells one embryo? Well, what we really mean is that these cells are aligned both physically and behaviorally in terms of anatomical space. They're aligned towards one single journey. They're all going to together cooperate and move from the state of a single cell to a morphology appropriate to a human or chicken or whatever it's going to be. And that's why we call it an embryo. It's really a story of alignment. But in fact, if you take that same blastoderm, and I used to do this with duck embryos when I was in grad school, you take a little needle and you put some scratches into this blastoderm, eventually they heal up. But before they heal, what happens is that every one of these regions doesn't know about the presence of the other, and it self-organizes into an embryo, into a whole organism. So in the end, you get here, so these are twins, you can get triplets and whatever. So the question of how many beings, how many cells, how many individuals are in a single embryonic blastoderm is not known in advance. It is not nailed down by the genetics. The default is one, but the capabilities here are anywhere from 0 to probably half a dozen or more. And the earlier you go in development, the more cells you can make out of a single embryo. So some fascinating issues here about cells having to decide where do I end and the outside world begins, which cells belong to which embryo, having lots of parallels that I don't have time to talk about now in terms of both individuation and dissociative disorders in cognitive systems, this idea of having a material, an excitable medium in which multiple self-organizing individuals can form. But understanding all of these stories, the genetic information is just the beginning. Here's another example. This time in behavioral space. So here's a tadpole. There's the mouth, the nostrils, the brain, the gut, and the tail. What you're seeing is that we've prevented the primary eyes from forming up here, but what Doug Blackiston did do is put an eye on its tail. And these eyes then, they make an optic nerve. The optic nerve sometimes synapses onto the spinal cord, sometimes not, sometimes it goes to the gut. But in most of these cases, these animals can see. And we built this machine that tracks their behavioral competencies with a visual behavioral trial. And yeah, they can see out of these eyes. Now, these eyes don't connect to the brain. The sensory motor architecture has been radically altered. Why doesn't it take additional rounds of evolutionary adaptation, meaning genetic change and selection, for this to work? Why does it work out of the box? Why does it have this incredible plasticity? And partly this is because what I'm going to emphasize in the next part of the talk is the problem-solving competency of what it is that evolution makes. It does not make, in almost all cases, it does not make genetically determined sets of phenotypes. It makes problem-solving agents that attempt to put together a coherent life story from the beginning, whether or not they're in the default mode of being a normal embryo or under these weird conditions that I'm putting these things in. You might also ask, by the way, just for fun, what happens to this eye? Because what we know is that this whole tail is going to disappear. The tail gets these signals to die, and eventually it gets smaller and smaller, and eventually disappears. So in the journey from tadpole to frog, the tail has to apoptose and die. What will happen to this eye? And we made some of these, and we had a kind of a poll going in the lab as far as what people predicted what would happen. If you're wondering, what kind of genetic data would you need to answer this question, right? I mean, all the genetics is normal; it's just a normal xenophage genome. What information would you need to answer this question? So what actually happens is that the tail gets the message to die. All of these cells self-destruct. But as that happens, the eye basically rides back to the end, growing the whole time, ignoring all of those messages, and ends up this beautiful eye on the butt of this frog. So again, that incredible plasticity, and it gets really wild. This is one of my favorite examples. This is a cross section through the lumen of a kidney tubule in a newt. So normally it's 8 to 10 cells that work together to build this kind of structure. But one thing you can do is you can make polyploid newts. So they have extra copies of their chromosomes. And when you do that, what you notice is so you see, here's some amazing things that you notice. This is Fankhauser's work back in the 40s. The first thing you notice is that 2N, 4N, 6N, 5N, these massive uh, uh, excess copy numbers of all the genes, you still get a perfectly normal nucleus. Not a problem. You have more genetic material, not an issue. The cells actually get bigger to accommodate the bigger nuclei. Okay, so that's the second thing. The cells actually adjust to this bizarre scenario. Uh, but also the newts that you get are the exact same size as normal. So in order to do that, what the system does is now fewer cells get together to make the same structure. So the third amazing thing is that it actually adjusts the number of cells to the new cell size. And then the most amazing thing of all is that when you make these truly gigantic cells, one cell will wrap around itself, leave a hole in the middle, and give you the exact same structure. So so first of all, this is an amazing example of top-down causation because in the pursuit of a large-scale anatomical outcome, different molecular mechanisms are getting called up. So in this case, cell-to-cell communication, in this case, cytoskeletal bending. In order to solve this problem, different molecular affordances, and you'll see I'll talk more about this when we get to the anthrobots, different aspects of the molecular hardware are getting called up to do their job. So think about what this means. If you're a newt coming into this world, you can't count on knowing or depending on how much DNA you're going to have. You don't know what your cell size is going to be. Other experiments show that whether we have a weird cell number, it doesn't matter. You can add cells, take away cells, you still get a normal newt. You have to be able to do all this, even if you can't rely on, never mind the environment, but you can't even rely on your own parts. And the genetic information that you inherited from your ancestors certainly is useful. It has all kinds of useful tools in it, but it is not by itself an accurate guide to your life and what you're going to have to do. So that creative on-the-fly interpretation of what you have is critical. And that ability, that flexibility, that reprogrammability gives rise to some amazing outcomes for us engineers. For example, here's an oak, and reliably, billions upon billions of times as you look around, it makes this nice, flat, green, reliable, extremely reliable outcome. And you might get the idea that that is what the oak genome knows how to do. The oak genome specifies, I think a lot of people would say that, specifies this exact structure. Except along comes a bioengineer, and it happens to be not a human bioengineer, it is a wasp. And what the wasp does is prompt these leaves, the various signals that the wasp is laying down, to build something completely different. Would you have any idea, if we didn't have the wasp showing us this, would we have any clue that these nice flat green cells that are so reliable actually have the ability to build something like this? Again, these are not wasp cells, these are plant cells. And this thing is eminently hackable. And if the wasp is doing this, what else might you be able to get these cells to do? What else is in their repertoire? If you thought that genetics specifies green leaves of a specific type, you would be very surprised to find that actually they can do this. Now, the thing is that better engineers make for better, more elaborate constructions. So if you look at the different types of plant galls in the world, bacteria make just basically a big featureless blob. Fungi induce little, sort of slightly better controlled structures. Nematodes, mites do a little better. Now you're in 3 dimensions, and then insects can actually do something like this. This is another plant gall. So what can we as bioengineers do when faced with a material that is so incredibly willing to use the genetically specified hardware that it has for novel purposes, novel journeys in the anatomical space and physiological space, and so on. A few more examples that I'm going to go through where we can start to see how much information is actually limiting from the standard genetics that we look at. I've spoken about these before. These are planaria, and planaria have some incredible properties. They are super regenerative, so every piece of this planarian will rebuild a perfect little worm. They are also cancer resistant, and they are immortal. So the asexual form of this animal shows no evidence of aging. They basically go on forever. And for a reason that I'll describe momentarily, they're extremely resistant to transgenesis. Now, one of the big puzzles here is that these animals, because they reproduce asexually, they split themselves in half and regenerate. That means that unlike us, who clean our our genomes, we don't pass on our somatic mutations to our offspring, right? We have a Weissman's barrier where if we get a mutation in our arm, our child doesn't inherit that. But in planaria, they do. So every mutation that doesn't kill the stem cell is going to be repopulated, in fact, magnified into the next generation as the offspring rebuild their bodies. So their genome is incredibly noisy. They bear massive amounts of mutation and so on. And so, what aspect of the genetic paradigm that we all learned in school, where the genome is really important, the genome determines what the cells are going to do, what aspect of that paradigm predicted that the animal with the noisiest genome, the one that accumulates all these mutations, is actually the one that's going to be immortal, extremely robust with respect to its ability to regenerate after injury, and cancer resistant? I mean, it's exactly backwards, right? There's nothing that would suggest this. And in fact, I think the key, and we've done a lot of, we can talk about this in the Q&A. We've done a lot of both computational and also bench experiments on this. Part of the reason that these guys are so resistant to transgenesis is that they don't take their genomes all that seriously. They can't, because they're functioning with an extremely unreliable hardware, and thus what evolution has done is put a lot of effort into an algorithm, an error correction algorithm that allows them to get the job done, even if and inevitably when the hardware is faulty. And that's why changing the genomes here doesn't really lead to anything. And the only known abnormal line of planaria, right? Like every other organism, mice, fruit flies, you can order fruit flies with weird, crinkly wings, and mice with bent tails and all that. There are no genetic lines of planaria except for the two-headed line that we made, and that was not genetic. As I'll show you momentarily, that was bioelectric. So, in going over all of these examples, I just want to be super clear that I'm not claiming that genetics is not useful. Of course, it's useful. Understanding your hardware is essential. Nor am I claiming, when I talk about the software reprogrammability, I'm not claiming that our conventional computational formalisms, Turing machines, the von Neumann architecture that your laptop runs on, and so on. I'm absolutely not saying that these things are sufficient for understanding life at mind. What I am claiming is that all the metaphors we use, and things like genes, pathways, are metaphors, just as memories, reprogrammability, and those kinds of things are. We want metaphors and conceptual tools that optimally facilitate invention and discovery. It's not enough to look at something that was discovered and look backwards and say, well, yes, of course, there's some kind of biochemical explanation that leads to it. Right, looking backwards is easy, and it's always going. If you zoom in far enough, it's always going to be biochemistry, not fairies, underneath, and underneath of that will be quantum foam or something. But that's looking backwards. The question is, can we develop formalisms that help us discover these things, right? And understand the incredible plasticity and the problem solving of this material that we're dealing with. So let's talk for a minute about the implication. What do we do now? Now that we understand that there's biology clearly needs more than just the hardware to figure out what's going on and how to take advantage of it. What can we do next? Well, the first thing I just want to mention, because I'm going to focus on bioelectricity, but it's not that bioelectricity is the only way to do these things. For example, biochemical circuits are also reprogrammable. You can take a look here. We found that gene regulatory networks, so just a few genes that turn each other on and off. That's it. No nucleus, no cytoplasm. You don't need anything other than a few genes that turn each other on and off. That kind of a system is already able to exhibit 6 different kinds of memory, including Pavlovian conditioning. You can do things like train them, and we're working on some protocols for drug conditioning and things like that. So Patrick Erickson's building these devices to exploit the memory of cells and tissues for specific pharmacological stimuli using the tools of behavioral and cognitive science. We can train these things. And so the biochemistry underneath within cells does this too. But I think there's something unique and interesting about bioelectricity. And what I'm proposing is a metaphor. And again, this is not to be the exclusive way to look at this. I think multiple metaphors are appropriate. But the one that I would like to use, the formalism, is this: that what the traditional way to look at it is here. People often refer to the DNA as the software of life and the cell as the hardware. I actually think we can do better than this. in understanding that what the DNA actually specifies is the hardware of life, all the proteins that cells get to have, including these ion channels. But one interesting thing about electrically excitable media is that once you once you produce a medium that has these channels that can open and close post-translationally, you can have self-organization, symmetry breaking, and all kinds of interesting properties, including memory and decision making, that happen at this layer. This is really incredible kind of hardware. Now, engineers understood this long ago. So here's a very simple, what's called a flip-flop circuit. And the cool thing about this the circuit is that it can store one of two states, a 0 or a 1, depending on which direction the current is flowing through the circuit. And once you apply a stimulus to one of these leads, so here you apply stimulus, that's it. Then you take your hands off the wheel, and now you've flipped it from 0 to 1, and it will, as long as the power stays on, of course, there's metabolism. As long as the metabolism is there to support it, it will keep that memory. And no amount of taking stock of the hardware is going to tell you what information is here. You can X-ray this thing and get an omics, right? So imagine doing omics of this. Okay, so I know what all the transistors, all the resistors are. I know what these two transistors are. I got them all. And that still doesn't tell you if the thing is at any given moment is bearing a 0 or a 1. This is the beginnings of very simple reprogrammability. And this now starts to connect to both neuroscience and connectionist paradigms in AI, where you have these kinds of networks that are easily implementable in electrical connections, but they do very interesting things, like what happens between the retina and the rest of the brain, where they start to pick out low-level features and then progressively generalize to higher and higher level features. And they can also do things like pattern completion and so on. These types of networks are incredibly powerful. So what we have is two coupled layers, each of which can generate and process its own information. They're certainly coupled because the genetics determines the ion channels, which in turn affects how the bioelectrics is going to go. And of course, the voltage then also turns genes on and off. So the whole thing is the cycle. But you really do have these two autonomous layers. And on a single cell level, you can always drill down, and we've done this numerous times in every paper. We have to show exactly how you get from a voltage, like first of all, what makes the voltage change, what's the transduction machinery, in this case, serotonergic signaling, what genes are downstream, what happens to the cells. You can track this pathway mechanistically very well, and we kind of tell these stories in all the papers. But the problem is this kind of mechanistic thing, which is required in a journal, is really just the start. It's not satisfying at all because there is no one-to-one mapping between the electric state and the genetic information. Many channels and pumps can give rise to the same exact voltage. And in fact, the same channel or pump can give rise to many different voltages depending on whether it's open or closed. You cannot see this on transcriptomics, on proteomics. This has to be measured in the living state. Now, of course, neuroscientists are very used to this because action potentials running through the nervous system do not require new proteins to be constantly made and taken away as the action potential goes down. We know that this rides on the physics of it. The hardware is already in place, and now it's excitable and so on. So we know that the protein level does not tell the whole story. And so in neuroscience, what's really good, and I mean they have many mysteries that remain to be cracked, but what they have established is this idea of a multi-scale investigation program where many different levels of description are appropriate. So yes, you can tell stories about the synaptic hardware and the way that the different ion channels give rise to specific shapes of potentials that move from cell to cell. But what they really want to do is to decode all this electrophysiology. So here's a zebrafish, living zebrafish brain. What they really want to do is decode all this electrophysiology and be able to extract the cognitive content. What are the memories, the preferences, the goals, the behavioral repertoires of this animal? And the idea is that all of those are somehow implemented in the electrophysiological network. And as I've mentioned here many times before, that system, the use of bioelectric networks to integrate information across space and time is incredibly ancient. So already bacterial biofilms do this. And every cell in your body has these ion channels. Most of them have gap junction connections to their neighbors, and we can we can start with the same kind of program, is to decode these pre-neural or aneural kinds of bioelectric dynamics and ask what are these to see the conversations that these cells are having with each other, or this is a frog embryo, these are explanted cells in a dish. Again, they have some very complex dynamics here. We do lots of computational modeling to try to understand how the excitable medium of cells actually propagates this information. And what we see is that not only do these electrical kinds of dynamics allow cells to cooperate within an embryo to build specific structures. So here's this electric face I've mentioned many times that in this movie by Danny Adams, where you can see here's where the animal's right eye is, here's where the mouth is going to be, here's where the placodes are going to be. All of this precedes and instructs the downstream gene expression and the anatomy of the face. But not only do these patterns serve as memories for normal development within an embryo, it turns out actually that these physiological dynamics allow propagation of information across embryonic groups. So here's this embryo in the middle is getting poked, and look, there's a calcium wave that allows all of these to know about it. The same thing happens here. Here, this one gets poked, and these two, which are not touching, these two find out about it. This is not just an epiphenomenon. Actually, groups of embryos, as we showed recently, resist teratogenic insults way better than individuals. So actually, the collective, it's kind of a wisdom of the crowds thing going on here. The collective knows how to make an embryo better than a single embryo knows how to make itself. And there are all kinds of rich dynamics here, both within the neurons and the non-neural cells that surround it. So we need to be able to read out these electrophysiological events. But of course, more important than that is the ability to rewrite them. In other words, to put information, to change information into this electrical network. And there are no fields, magnets, frequencies, waves, radiation, nothing like that. We basically manipulate the native interface, so these ion channels and gap junctions that the cells are using to hack each other. So all the tools that we appropriate from neuroscience work here. So pharmacology, optogenetics, mutating these channels if we want to see what the channel structure is contributing. That's how we put information back into the system, and that is how the cells control each other's behavior too. So let me show you a few examples that really address that main theme, going back to this genetics and what else. So one thing, and this is the work of Sherry Au and Vaibhav Pai, who show that if you try to recapitulate that little bioelectric eye spot that I showed you in the electric face, and you try to recapitulate that pattern somewhere else by the injection of, for example, potassium channel RNAs in cells that are going to give rise to other body regions, you can get eyes in other locations, right? And these eyes have lens, retina, optic nerve, all the normal stuff. In addition to showing that bioelectricity is instructive, it also shows something very interesting. If you look at the developmental biology textbook, what it'll say is that only the tissues up here in the anterior director are competent to be eye. Okay? They're saying that the tissues back here are incompetent to make eye because they were prompting it with the so-called master eye gene, PAC6. If you use PAC6, it's true. Injection of PAC6 only induces ectopic eyes up here. But that limitation of competency wasn't on the part of the tadpole or the cells. It was a limitation on the competency of us as bioengineers to be communicating using the wrong level of interface. If you stick to the genetics, you don't make eyes back there. If you go to the bioelectric state, you can make eyes probably anywhere, in fact, in the animal. So you have to be really careful when we assign competencies to these things. We ourselves are kind of taking an IQ test as far as what level of what interface of communication have we found, and have we found the right one. This is another example. This is also done by Pai, where what he showed is that embryos with incredibly damaging brain defects induced by a Notch mutation. Okay, so Notch is a really important neurogenesis gene. You mutate Notch, the forebrain is missing, the midbrain and hindbrain are a big bubble. These animals have almost no behavior to speak of. They're profoundly affected. Actually, we had a computational model made by Alexis Pitak who predicted that if we were to just activate a certain channel called HCN2, we could restore the bioelectric pattern that would force normal brain pattern. And in fact, we did that, and here it is. The brain structure is normal, gene expression goes back to normal, and their behavior, their learning rates become indistinguishable from controls. The mutation is still, the mutated notch protein is still here. But the phenotype is, that's not what drives the phenotype. It's the bioelectrics that drives the phenotype. So at least in this case, and I'm not claiming all cases, but at least in this case, and probably many, some hardware defects are fixable in software. So for the same reason that with the same genome under various perturbations, as I showed you before, the system can adapt. We can now address this bioelectrical interface to fix scenarios in which actually there is a permanent hardware defect, such as this notch mutation. This is another example of the same thing, and I've shown you this before. These planaria, this is our abnormal strain of planaria. They were made to be two-headed by rewriting the bioelectrical circuit that normally says one head, one tail. We can create these two-headed animals. If you cut them, they continue to be two-headed. There is nothing genetically wrong with them. If somebody were to sequence it, they would not find the speciation event that they would be looking for if they thought that these were some kind of different lineages. The question of how many heads should a planarian have is simply not nailed down by the genome. The default is, like a lot of hardware, under default conditions, it will do the same thing reliably. But that is not the only thing it can do. And it has that circuit has this amazing property of memory, such that once you've shifted it to a two-head state, it holds. And even small fragments of this thing will contain that information until you set it back. And we actually do know how to set it back now. Not only can you control so we're controlling the number of heads here and saying that does the genetics predict how many heads you're going to have? So you cannot, in fact, scan the genetics and tell how many heads this animal is going to have. Can you tell what kind of head shape is it going to have? Well, what you can do is you can take this species and cut off the head, again, perturb the bioelectrical network, and it can give rise to flat heads like a B. felina or round heads like an S. mediterranea. These are about 100 to 150 million years evolutionary distance. Okay? And not only the shape of the head, but also the shape of the brain and the distribution of the stem cells become like these other species. So in that anatomical morphospace that all bodies navigate, there are different attractors. Some species, so some genomes, make hardware that by default will end up visiting and staying in these other attractors. But actually you can push other species hardware to visit them as well. And this one is perfectly happy to go and kind of land in these other areas of anatomical space. So again, that slippage, right, between the specification of the genetic specification of the hardware and what the actual phenotypes might be, that slippage is massive, and there's a really important layer in between. In the case of cancer, so here's what a normal cross section through a tadpole looks like. Here are the melanocytes. Small number of nice little round pigment cells. This is on its way to metastatic melanoma. So these pigment cells have gone crazy. They have hyperextended. Look at the difference between this and this. So they acquire these incredible invasive morphologies. They dip down into the neural tube, here into the brain. They're metastasizing all over the place. They fill up the blood vessels. So they hit the blood vessels and just like melanoma likes to do. There is nothing genetically wrong with this animal. What we've done is temporarily disrupted, using a particular chloride channel, we've disrupted the communication between a certain cell type called instructor cells and the melanocytes that those instructor cells normally keep kind of suppressed. And when you interfere with that, the melanocytes go crazy, and they do this. And eventually, this thing will turn on a lot of cancer-associated genes, associated with epithelial mesenchymal transition and so on. But it isn't because there was anything fundamentally wrong with the body. There were no oncogenes. There was no carcinogens. There was no DNA damage. Nothing like that is necessary to induce this kind of cancer phenotype. Let's look at the flip side. If you do inject human oncogenes into these things, they will eventually make a tumor. But the reason they make a tumor is because the first thing that oncogenes cause is an electrical dissociation of cells from their neighbors. So the cells depolarize, they disconnect from their neighbors. You can see this happening before the tumor emerges. In fact, you can see some other regions here that if you were a surgeon, you want to keep an eye on. What you can then do is target this process, and as we did here, and this is the work of Brooke Chernet, where he injected the oncogene, and we're talking about nasty KRAS mutations and p53 mutations and so on. And instead of a tumor, you'll get a perfectly normal skin or muscle and all that. This is the same animal. So the oncoprotein is labeled in red. It's blazingly expressed. It's all over the place. We didn't kill the cells. We didn't shut off the oncoprotein. We didn't get rid of the oncoprotein. We didn't repair the DNA. If you sequence this thing, you will in fact make a prediction that it should have a tumor because you're catching this oncogenic mutation. But there is no tumor because what we did was co-inject an ion channel that keeps the cells connected to their neighbors, and it's the electrophysiological pattern that the cellular collective has, which keeps it focused on making both muscle and skin and all the organs, as opposed to the single cell behavior that we see here in metastasis when those connections are broken. This is what drives, not the genetics. And so this is a kind of weird, you could say in this analogy that we've been pushing for years, which is that there's a real symmetry between the problem-solving capabilities of groups of cells in morphogenesis and those in the brain during conventional behavior, is that cancer, on that view, is literally a dissociative identity disorder of the morphogenetic intelligence. And what we're doing here is a kind of reintegration therapy that takes advantage of the plasticity to reconnect them and have the decision making take place at the level of the collective, not of the individual cells. So, just a couple of things to finish up here. If I were to show you something like this, and this is the work of Gizem Kamushkaya, a PhD student in my group, if I were to show you this and ask you to say, what do you think this is? So from its behavior and morphology, you might guess that this is something I got from the bottom of a pond somewhere. If you were to sequence the genome, and so based on that, you might make some predictions. You've seen some primitive organisms swimming around like this. It's covered in cilia, so that's how it moves around. It has these little hairs that row against the water. And looking at this form and function, you might make a prediction on what the genome is going to look like. It will probably look like one of these other organisms. In fact, it turns out this is 100% Homo sapiens genome. These are anthropods. We make them by taking epithelial cells from adult patient donors. Most of these are elderly donors that give biopsies of their tracheal epithelium. We put these cells into a particular environment that we've established, and they self-assemble into this novel multile little shape. This is not like any normal stage of human development. So you'd be very hard pressed to guess the genome from observing this, and you'd be very hard pressed to go from the human genome and having seen human developmental stages to say that anything like this is possible. Now, could you predict the transcriptome? You know the genome, fine. The genome is a Homo sapiens genome. Can we at least say what the gene expression is like? Well, it turns out that these anthropods, on the order of about 9,000 genes, are differentially expressed from their tissue of origin. Okay, so here's a volcano plot of a transcriptomic study that we did. All of these genes are differently expressed, about half the genome. Remember, there are no synthetic molecular biology circuits in there, so there's no symbio, there are no scaffolds, there are no weird drugs or mutations or anything else. These are completely normal adult patient cells. And just because they now have a different lifestyle, they're in a different environment, they're having a different life. The patient may or may not be alive. The anthropods have about 5 weeks of life like this, running around and doing their thing. And that novel lifestyle has enabled them to dip into their genome and radically rearrange the transcriptome. Okay, so there are thousands of genes that are now different, and this preprint that kind of goes into depth about what it is that they're actually what kind of genes are these. Well, one interesting thing is that if you and this is Leo Lopez, our bioinformatician, who actually did some phylostratigraphy on these genes and asked what are the ages of these novel genes that they're turning up or down. And it turns out that actually most of them predate the eukaryota. They're ancient. So what these things have done is by finding themselves in this novel environment and having kind of a new 5 weeks of new life, is they've most radically remodeled the very most ancient genes that actually predate eukaryotes. Now, what are they doing during their life? Well, here's one thing they're doing. So if you make a lawn of human neurons on a petri dish. So here they are. These are iPS-derived human neurons, somewhat similar to peripheral neurons. And we take a scalpel, we put a big scratch through the middle, so it's a scratch wound assay. We let the anthrobots in. They sort of run down these wounds, and then what they can do is they can pick a spot, they can land, they can uh, sort of stick together. They can merge together. We call this a superbot cluster. There's probably 10 or a dozen of them here. They stick together, and then they start doing something very interesting, which you can see when you lift them off. 4 days later, if you pick them up, what they were doing here is knitting the neural damage across the gap. What they do is they sit there and they connect the neurons on either side of the gap. Now, what genetics experiment would have led to this discovery? What would you have, how would you have possibly guessed without trying this that genetically normal, unmodified adult tracheal cells that sit in your airway for long periods of time quietly, wafting particulates out of your lungs, that they would be capable of forming a new kind of life form with a new morphology, radically changed transcriptome, new behavior, and the ability to repair other tissues. I'll also point out that this is the first thing we tested. This isn't experiment 82 out of 1,000 things we tried. This is the first thing we tried, which suggests to me that they probably have all kinds of other capabilities that we haven't even scratched pardon the pun, scratched the surface of. So this kind of plasticity and the fact that this was not the result of a genetic screen. This was not the result of genetic modifications. We can keep thinking about this idea of what kind of formalism facilitates this kind of discovery. And it was not made by investigating the genetics. It was made by thinking about the kinds of agential plasticity that cells have in their ability to organize new lifestyles in new environments. So the very last thing, just for a couple minutes, I'm almost done. I want to cover something pretty strange, which are some ideas beyond genetics and environment. So one thing that we've done is some interesting computational studies around what does it mean for evolution that your material is smart, that is evolving over a very simple mapping. So you mutate your genome, and then you select on the phenotype, and that makes for very kind of well-known evolutionary dynamics, and people do evolutionary algorithms and so on. But if you put in that middle layer, which is very rare in this field, when people simulate these things, they don't usually put in that layer. But when you put in that layer, that problem-solving physiological layer between the genetics and the actual behavior, something very interesting happens. You end up with an intelligence ratchet, because that competency, the ability to make something functional even though your genetics have been tampered with, which I've shown you many examples of, what that does in effect is hide information from selection. It makes it hard for selection to see the best genomes, because really good animals, in fact, may have problematic structural genomes. You just can't see it because things got repaired. And I've talked here before about the ability of faces to fix themselves in morphogenesis, in metamorphosis rather, and so on. So what happens is that you end up with this interesting ratchet where, because evolution can't see the best genomes, what it does is spend all of its time cranking the competency machinery, which in turn hides the genome even more, makes it even harder to do that, and then that positive feedback really enables this rise of competency, and you end up with animals all along the spectrum. And you don't know why different lineages stop at different places, but for example, C. elegans are incredibly hardwired. Every cell is numbered. Every cell is exactly what it's supposed to be. The lineage relationships, and that's it. And then planaria are all the way on the other end, which are crazy plastic and amenable to all these things. And then mammals and amphibia are somewhere in between. But there is this really important thing, which I think in the end results in many different kinds of intelligence. This idea of an unreliable substrate, and the fact that you have to, from the word go, you can't really rely on what your molecular information is. You have to reinterpret it, whether you're an anthrobot or a xenobot or a newt with the wrong amount of genetic material or whatever you are. You have to develop the story from scratch, and that leads to this positive feedback. So now we come to an important question. When I show you these kind of crazy competencies, these new forms, new behaviors, new functions, where do they come from? So one way to think about this, and I think a lot of people think about it this way, is that they're emergent. And what they mean by emergent is that they're surprising. In other words, yes, there were low-level rules of chemistry or whatever else, and sometimes they do these things. They often focus on complexity and unpredictability, but it's far more than that. It's not just complexity that's emergent. It's actually basal cognition. It's the ability to solve problems and to make a coherent, adaptive story in some space. So okay, so emergent problem-solving capacities. Um, and so now we have a choice. We have a choice to make. We can say that there is this random grab bag of facts that hold. Things about networks, like the kind of stuff that Stu Kaufman studies, or all these other things. They're emergent in the sense that they come up from time to time. They're unpredictable. They're not obvious from the hardware description, and so we just have to sort of catalog them when they emerge. We are surprised. We catalog them, and there it is. That's one way to look at it. Another way to look at it, which I think is much more fruitful, is what mathematicians do about these things. And a lot of mathematicians believe that they are not inventing mathematics; they're discovering it. In other words, there is a Platonic space of truths, and it's not just random stuff that occasionally shows up in the catalog, but it has a metric, it has a structure to it. In fact, you can see that they make this thing called a map of mathematics, where different mathematical, the truths of number theory and topology and all these things are actually arranged in a certain way that you can actually investigate it in a systematic fashion. And so my proposal is that we take this very seriously, and that it's not just the truths of geometry and number theory and things like that that live in this option space, or latent space, as the computer scientists call it, or the Platonic space, as the mathematicians call it. But there are actually other patterns there that are relevant to biology that evolution exploits, thoroughly exploits by taking advantage of them, and we take on the task of systematically exploring this latent space, and that means that the things that we make here, so of course standard embryos, but all of the bioengineered stuff, all the biobots and the organoids and the crazy constructions that bioengineers make, are exploration vehicles. They are ways to peek into this latent space to see what other patterns exist around the conventional patterns of normal embryos and so on, and to start to understand it and develop a map of that space so that we can rationally take advantage of it for biomedicine for other purposes. In other words, if physical constructs that we make are basically pointers into that space because they avail themselves of all sorts of affordances that are not determined by physical laws. For example, the truths of number theory, the things from computer science, like the fact that the NAND gate is universal, and all these kinds of things that would not change if the laws of physics were different, and those things would still hold. We should be exploring that space. I want to just give you a very simple example at the very end. As biologists, we like, when we say where does information come from, we like two things. We like genetics and we like environment. Where else could it possibly come from? So here's an example. This thing you're looking at here is called a Halley plot. It is a type of fractal. It's a very simple algorithm that takes a simple equation in complex numbers. This is literally it. It's only 1, 2, 3, 4, 5, 6 characters. That's all it takes. And this is the pattern that it actually encodes. And if you can make movies out of this by modifying these numbers slightly as frames, and you can see here, this is what's baked in. This structure, and it happens to be very biological looking, but that doesn't necessarily matter. Just the incredible order here is all of that comes from this little seed right here. If you were able to go backwards, which we can't, but if you were able to take this and compress that down to something like this, can you imagine the degree of compression? We don't have anything remotely like that. This is a generative seed that inside of this is buried all of this stuff. Where does that come from? There is no story to be told anything remotely like genetics or environment, and I would argue against this idea that we just need to accept that it's just the fact that holds. It's just one of these things that this is the pattern that lives here, and when we come across these, we can be surprised and catalog them. I think the fact that these patterns exist is a call to explore them, and biology and evolution uses them extensively. This is something that we're doing in our lab, is in collaboration with Don Ingber's group at the Beast, is to create systems that involve kind of robot scientist platforms, so robotics and AI, to have a cycle of discovery and to be able to map out that space in a way that is not mapping information at the level of genetics or omics or anything like that, but it's actually mapping the patterns, the protocognitive patterns that these systems can embody in terms of problem solving in these various spaces. So I'm gonna finish here just to point out that we want a level of description that facilitates discovery, and that the mapping between the genetic data and the phenotypic outcomes is really not strong at all. It is not a blueprint in any sense. Instead, it's a parts list for a self-assembling, reprogrammable, problem-solving agent. And the formalism that is most predictive, biochemistry, emergence, complexity gives us some of it, but the future, I think, belongs to tools from the emerging field of diverse intelligence and some tools from computational and neuroscience. I've talked many times about bioelectrics and why that's a really good interface to this, but there are going to be many, many more. And I think in the end, we're going to have to redefine this question. Where does the information come from? This idea of where is information we don't even have a good way to understand these answers yet. So I think we need to go beyond the mysterianism of emergence and to explore that space explicitly. And when we do that, then we can have this other part of biomedicine, which is all the top-down stuff here, which is the ability to behavior shape, to train cells and tissues, to have agential implants like anthrobots that are made of your own body cells going into your body to fix you don't need immunosuppression, to repair your own tissues, and all kinds of things that I've spoken about before in terms of electroceuticals and so on. And all of this is described in detail in these papers. And I'm happy to send out any of these things. So just to thank the people who do all the work. So these are the amazing students and postdocs who work in our lab. Lots of collaborators. So these folks did all the work that I showed you today. Lots of great collaborators. Our funding support, including especially Guy Foundation. So thank you for that. Disclosures. There are 3 companies that have supported some of this work, and always have to thank the model systems because they do all the heavy lifting for us. So thank you, and I've gone over. So I'll take questions.
Mike, thank you. I suspect a number of our participants are as stunned as I am in what we've just seen. And we're most grateful that you've tried this presentation out on our platform first. Can I just suggest if people want to ask questions, put their electronic hands up. You see Alistair's there straight away. Alistair.
Thank you, Mike. That was brilliant. Absolutely fantastic. Every time I learn something new and think differently, slightly differently. Something just occurred to me. Is it possible that through evolution, molecules have evolved which kind of can adapt to stress, the system you're talking about? They're a bit like morphoceuticals. And I'm thinking in particular, you mentioned there these anti-epileptics, but you've also got a lot of natural plant products.
Yes.
We think of them just having specific targets, but what if they're altering the whole electric field as an adaptive response when something's gone wrong? It gets back to our cannabidiol question. We talked about this a long time ago, but potentially these seem to help the plant survive in certain situations when it's damaged. So could these molecules somehow be reorganizing, maybe by controlling ion channels, which brings this as a stress response to explore that phase space, if you were to bring back. Is this something which I think you kind of almost mentioned it at the end, but just a thought that actually this is what they could be doing.
Yeah. No, I think that's absolutely a possible scenario. We've done a bunch of work on serotonin and the ancient pre-neural roles of serotonin, so that's there for sure. But we are also now getting into, and this is some of the stuff that Pi and I are going to be doing soon, is to use some of the psychedelics and those kinds of compounds to coordinate large-scale stress response decision-making. We've done experiments before where we've made embryos hallucinate in morphogenetic space, and it's very interesting. You can use SSRIs and other things that distort perception and decision-making, and you get in anatomical space exactly the same kinds of things that we get in 3D behavior when animals take them. So I think that's a very promising direction for research.
Yeah, because it kind of explains how these molecules can have multiple effects. They can be anti-cancer, anti-inflammatory.
Absolutely.
and even longevity inducing. You kind of think, well, how are they doing all of these things? And actually, what you're looking at, what you're suggesting, seems to be one obvious way.
Just had a couple of thoughts. One is that the first third of your talk is almost a syllogism proving DNA is not a blueprint, or it is not the blueprint. That's almost like the phrase you want to start out with. So what I'm thinking is, how do you communicate this to most ordinary biologists? And that would be one thing. The other is your flip-flop jogged my memory, and I have a suggestion. So the way biologists think is that one protein's kicking another one, phosphorylated or whatever, you get some hormone or whatever. And as soon as that stimulus is gone, you go back to normal. And what your point here really is about the programmability is, no, what you can do is have a permanent, you have a transient signal that makes a permanent change in state. And now that looks like a computer program or a computer running a program. And then biologists may get it.
Yeah. I think that's exactly right. And there's a couple of funny things to say about that. One is when we are so we have two papers now where we show that transient stimulation of a gene regulatory network circuit induces permanent changes in the way that it will respond to future stimuli, like, for example, in associative learning. So these things can actually do associative learning. But in our models, we do not let the physical construct change. In other words, you don't change the weight, the connection between the nodes, the strengths, nothing. Physically, nothing changes. Okay, it is not a model of synaptic plasticity. Absolutely nothing changes in the hardware. All of the memories and the dynamical systems aspects of it. Both papers, the reviewers were just, it was just such a, they were saying, show us, put your finger on where the memory is, right? They were saying, if the hardware isn't changing, where could the memory possibly be? That it was like such a barrier to so I think that's a primary thing. And then another issue is that there's a whole other community that I'm sure the people here know, which, okay, there's the conventional molecular biologists who want all this to be billiard balls and mechanism and all of that. But then there are all these organicist thinkers who get really upset when we use phrases like programmability, algorithm, software, because to them this is destroying an important binary distinction that they fought for hundreds of years to preserve, which is the majesty of life and the fact that you shouldn't reduce it to machine metaphors. So both of those things are somewhat unpopular in different crowds, and it's that walking that midline to just say that, look, we need conceptual paradigms that lead to novel discoveries, not to prop up old philosophical categories.
Yeah. So I would suggest losing the word memory, because that has so many connotations, and just replace it with change in state, which is much more neutral. The other thing that it sounds like you're running up against, so I got invited to give a talk about how do you get revolutions in science, so I've been thinking about that this week. The other thing is that the way science funding is set up, where you have to know kind of what you're going to find and how you're going to do it, is okay for reductionism, because you can always say, okay, I know about this protein, now I can look at the N terminus instead of the C terminus. And, but as soon as you try to do the opposite, where you're building up, now you're trying to pull two parts together, and there's no guarantee that those are going to be the right two parts. And so there's fudge sections, and evidently what you're saying, the publishers or manuscript reviewers just don't get it. I'm not sure what the cure for that is. Although I found out, I wondered how Einstein got his paper published. It turned out in those days there was not peer review, and the editor of that journal happened to be Max Planck.
Look at the interaction of the molecular or cellular agents. Do you think about communication between those agents as asynchronous or synchronous? And to the extent you think about that at all, do you think that the synchrony, to the extent it looks to be observable, or asynchrony, is a function of propagation of electrical signal across the various gap junctions and ion channels? How do you think about that?
Yeah, great point. The individual actions are asynchronous. However, there are emergent wave fronts and there are definitely emergent synchronous phenomena that we observe. So I think underneath there is no well, at least we haven't found. I mean, okay, there are circadian rhythms and hormonal rhythms and things, but typically it's not baked into there's not an explicit mechanism for, okay, let's all synchronize together. It's the fact that if you do certain things, you will end up with a metastable phenomena like solid, almost like gliders in the cellular automaton, things like this, where you do in fact have a very nice smooth progression where things look synchronized. But I think most of that is not baked in at the bottom.
And then, if I may, just another point. I wonder whether, in describing these agential materials as vehicles for exploring photonic latent space, there's not a bridge to Carl Friston's active inference framework there, in that perhaps the organism is modeling its environment, or even if we think about mitochondria and Peter Mitchell's original hypothesis, looking at the organism responding to or as a phase shift across a membrane, whether we can't view the organism as seeking to exercise a repertoire of responses to a stimulus in an